Hi there! You probably have heard by now why it's so important to solve a membrane protein structure. This is because membrane proteins make up more than 30% of our proteins and approximately 50 to 60% of our core and drug targets. And you have probably heard how protein structures are generally being solved by X-ray crystallography, which requires a well-diffracting crystal. But do you know that obtaining this crystal is still a bottleneck in the whole process? Well, today you are going to learn about it. My project is aiming for a solution to tackle this problem with experimental phase diagrams. But first of all, let's look at how we produce membrane proteins. Advances in biotechnology have helped us to rationally produce membrane proteins by recombinant expression. We do that by inserting the DNA sequence of our desired membrane protein into a plasmid, a circular DNA molecule used by bacteria and yeast cells to produce proteins. The protein will then be extracted by physically or chemically breaking the cells. And by making use of the chemical and physical characteristics of the membrane protein, we can design efficient purification protocols which separate the membrane protein by its charge and size from other components in the cell. During the whole process, we have to consider that membrane proteins, as the name suggests, are located in the membrane. The membrane is a phospholipid bilayer. This means membrane proteins require detergents, which mimics the cell membrane to keep the protein happy in its new environment in solution. Once you have your membrane protein solubilized in detergent, which we call a protein-detergent complex, the routine workflow is testing it against many different chemical conditions to find out which one will lead to crystallization. This high-throughput approach is called screening. While screening itself has been speed up by the development of crystallization robots, which can set up hundreds of crystallization conditions in a short amount of time, the success rate from screening experiments is not predictable. Why? We have not yet obtained a comprehensive understanding on the role of each component during the crystallization process, which facilitate crystal nucleation and growth. What we know is that crystal contacts, a connection between the molecules, are required to form a crystal. So aiming for the understanding on how to fine-tune the relevant parameters to establish these crystal contacts is essential. And because we are lacking this knowledge, we apply screening experiments to find the right chemical cocktail for crystallization. This involves looking at hundreds of conditions. Here you see an example of how screening results could look like. The crystals are so small in micrometer sizes that we need a microscope to look at it in most of the cases. Manually taking notes of every condition takes hours and hours. What happens in reality is that you probably only have the time looking at the crystal hits and dispose of every other result. By doing this, we would not be able to understand where and why crystallization occurs in the bigger picture. What we aim for in part of my project is trying to find an efficient way to analyze those screening results and link them to a crystallization phase diagram. The basics of a protein crystallization phase diagram include the undersaturated and supersaturated zone. It's also known that optimal growth condition exists in the metastable zone, and that the nucleation zone is where we can observe crystal formation. This diagram would give us a better idea on where to find more crystals and how to optimize membrane protein crystallization.